guys, let's learn about protein synthesis. Let's go!
We all know what DNA is, especially when it comes to crime related to murder or killing. The news often reports, "We found the DNA of the main suspect." With DNA, we can identify who the killer is. But have we ever felt curious about how DNA can produce characteristics? Let's take eye color as an example. The genetic information for our eye color is contained in our DNA. The pigment in our eyes result in our eye color. To have that pigment, you need a gene within your DNA that codes for proteins responsible for producing that pigment. So, I will explain how our DNA result in the making of proteins. This process is called protein synthesis. Synthesis essentially means to create something. So, protein synthesis is the process of creating proteins. We might wonder, what's the significance of proteins? Proteins indeed play a big role in our lives, even though we might not notice it. They do a variety of things. They play roles in transportation, provide structural support, act as enzymes for various biochemical reactions, and offer protection to the body among other functions. Proteins are important for us to live. Interestingly, we are making proteins right now as we sit down and play with our smartphone. This process happens in our cell. So, let's return to our DNA and its role in all of this. All our cells, with a few exceptions, contain DNA located in the nucleus. Some DNA is non-coding, and some DNA forms genes that are not activated. But let's focus on the genes that encode active proteins. How do we extract the information from these genes out of the nucleus so that cell can start producing the proteins they need? Allow me to introduce the amazing work of RNA. In brief, RNA is a nucleic acid like DNA, but there are some differences. Its role in protein synthesis is also massive.
First, you need to remember, in protein synthesis, we have two major steps. The first step is transcription, and the other step is translation. So, in transcription, we transcribe DNA into a message, and since DNA is in the nucleus, we are doing the transcription in the nucleus. During transcription, a piece of DNA that codes for a specific gene is copied into mRNA, which means messenger RNA. In this process, RNA polymerase is an enzyme that is responsible for copying a DNA sequence into an RNA sequence. The message will be sent out of the nucleus to the ribosome. Ribosomes make proteins, and ribosomes are made of rRNA, which means ribosomal RNA. Ribosomal gonna make a lot of protein and send it to the next step, which is translation.
In the cytoplasm, we have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer of a protein, like building blocks for a protein. Since we are making proteins, we're going to need these amino acids to build it. If we have a bunch of amino acids, you can build a protein. So, it's the tRNA that is going to bring the amino acids together to make protein. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the messenger, is so important, because it's going to direct which tRNAs come in and therefore amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acids. When tRNA brings the amino acids, it reads the bases, represented by letters in 3, here in the mRNA. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All the tRNAs that have the anticodon UAC will be carrying an amino acid that is called methionine. A tRNA with a UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transferred the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind the amino acid. That is the first amino acid before looking at the next codon. The tRNA will transfer that amino acid and eventually leave where it can go pick up another amino acid. These amino acids are held together by peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished.
Last but not least, protein synthesis is very important to living things. It creates structures that are needed for both individual cells and body. It also allows cells to produce these necessary components in response to changing the needs such as growth, repair, immune response, and regulating psychological processes. So without protein synthesis, life as we know couldn't exist. That's all from us. Thank you for watching.